Dear students, Assalamu alaikum. In the last lecture, we started the topic of tubular reabsorption. The objectives of today's lecture are to learn about tubular reabsorption of water and solutes and reabsorption and secretion in different parts of nephron. So we continue with the tubular reabsorption of water and solutes. Passive reabsorption of water by osmosis. When solutes are transported out of the tubule by either primary or secondary active transport, the solute concentrations decrease inside the tubule and increase in the renal interstitium. This creates a concentration difference that causes osmosis of water. The osmosis of water is mainly dependent on the permeability of the tight junctions between the epithelial cells than the osmotic gradient. In the proximal tubule, the water permeability is high and water is reabsorbed as rapidly as the solutes. As water moves across the tight junctions by osmosis, it can also carry with it some of the solutes, and this process is referred to as solvent drag. In the more distal parts of the nephron, tight junctions are much less permeable to water and solutes, and epithelial cells also have a decreased membrane surface area. Therefore, water cannot move easily across the tight junctions of tubular membrane by osmosis. However, water permeability in the distal tubules, collecting tubules, and collecting ducts can be high or low depending on the presence or absence of antidiuretic hormone. Reabsorption of solutes by passive diffusion. When sodium is reabsorbed through the tubular epithelial cell, negative ions such as chloride are transported along with sodium because of electrical potentials. Transport of positively charged sodium ions leaves the inside of the lumen negatively charged compared with the interstitial fluid. This causes chloride ions to diffuse passively through the paracellular pathway. When water is reabsorbed from the tubule by osmosis, the chloride ions are concentrated in the tubular lumen. Reabsorption of chloride ions then occurs because of its concentration gradient. Chloride ions can also be reabsorbed by secondary active transport, and it involves co-transport of chloride ions with sodium across the luminal membrane. Now let's study the reabsorption of urea ions. As water is reabsorbed from the tubules by osmosis, urea concentration in the tubular lumen increases, just like chloride ions. Reabsorption of urea ion occurs because of its concentration gradient from the tubule into the epithelial cell. However, urea does not permeate the tubule as readily as water. Therefore, in inner medullary collecting duct, passive urea reabsorption is facilitated by specific urea transporters. Only about 1/2 of the urea that is filtered by the glomerular capillaries is reabsorbed from the tubules. The remainder of the urea passes is excreted in the urine, and 90% of waste nitrogen is excreted by the kidneys as urea. The figure highlights the mechanisms by which water, chloride, and urea reabsorption are coupled with the sodium reabsorption. Reabsorption and secretion in different parts of the nephron. We now discuss the different characteristics of the individual tubular segments that enable them to perform their specific functions. Let's first concentrate on proximal tubular reabsorption. Normally, about 65% of the filtered load of sodium, chloride, and water are reabsorbed by the proximal tubule before the filtrate reaches the loop of Henle. Proximal tubules have a high capacity for active and passive reabsorption. This high capacity of the proximal tubule for reabsorption results from its special cellular characteristics. So, what are these characteristics? The characteristics of proximal tubule epithelial cells are: number one, they are highly metabolic, containing large number of mitochondria. Number two, the cells have extensive brush border, intercellular and basal ion channels, thus providing extensive membrane surface area on the luminal and basolateral sides, that is around all sides of the cells. The surface of the epithelial brush border also has protein carrier molecules which moves sodium ions by, number one, co-transport with the amino acids and glucose, and number two, by counter-transport while secreting hydrogen ions. Now, keep in mind here that there are some differences in the mechanisms by which sodium and chloride are transported through the luminal side of the early and late portions of proximal tubular membrane. In the early or the first half of the proximal tubule, sodium is reabsorbed by co-transport along with glucose, amino acids, and other solutes. In the late or second half of the proximal tubule, sodium is reabsorbed with chloride ions, as little glucose and few amino acids are left behind to be absorbed. They have already been absorbed earlier. The second half of the proximal tubule has a high concentration of chloride. The higher chloride concentration causes its diffusion from the lumen through the intercellular junctions into the renal interstitial fluid. Smaller amounts of chloride may also be reabsorbed through specific chloride channels in the proximal tubular cell membrane. Now, in addition to the reabsorption The proximal tubule is also an important site for the secretion of number one, organic acids and bases such as bile salts, oxalates, urates, and catecholamines. Number two, for secretion of toxins. Number three, for secretion of drugs like penicillin and salicylates. And number four, for secretion of para-amino hippuric acid. This figure shows the cellular ultrastructure and primary transport characteristics of the proximal tubule. As discussed earlier, the proximal tubules reabsorb about 65% of the filtered sodium, chloride, bicarbonate, potassium, and all the filtered glucose and amino acids. The proximal tubules also secrete organic acids, bases, and hydrogen ions into the tubular lumen. Now let's study transport in the loop of Henle. The loop of Henle consists of three functionally distinct segments: thin descending segment, thin ascending segment, and thick ascending segment. Out of these, thin descending and ascending segments have thin epithelial membranes with no brush border. I repeat, with no brush borders. The thin descending segment is highly permeable to water. I repeat, the thin descending segment is highly permeable to water. And about 20% of the filtered water is reabsorbed from the thin descending limb. The thin descending segment is moderately permeable to most solutes, including urea and sodium. However, it does not reabsorb calcium, bicarbonate, or magnesium. This figure shows cellular ultrastructure and transport characteristics of the thin descending loop of Henle. This part is highly permeable to water. It has few mitochondria and little or no active reabsorption. Thin ascending segment is completely impermeable to water, and it has much lower reabsorptive capacity than the thick segment. Okay, now let's study the transport in the thick ascending segment of the loop of Henle. I repeat, thick ascending segment. The thick ascending segment has thick epithelial cells that have high metabolic activity and are capable of active reabsorption of sodium, chloride, potassium, calcium, bicarbonate, and magnesium. Let's have a look at this figure which shows cellular ultrastructure and transport characteristics of thick ascending loop of Henle. This part reabsorbed about 25% of the filtered loads of sodium, chloride, potassium, calcium, bicarbonate, and magnesium. This segment also secretes hydrogen ions into the renal tubular lumen. The thick ascending segment also has the presence of sodium potassium ATPase pump in the basolateral membranes, which maintains low intracellular sodium concentration, providing two functions. Number one, it provides a gradient for movement of sodium from the renal tubular fluid into the interior of the cell. And number two, it provides potential energy for another co-transporter protein called 1 sodium 2 chloride 1 potassium co-transporter. In the thick ascending segment, the movement of sodium across the luminal membrane, the apical side of the membrane, is mediated by 1 sodium 2 chloride, 1 potassium co-transporter. And thick ascending segment is also the site of action of the loop diuretic drugs, which inhibit this transporter protein called 1 sodium 2 chloride 1 potassium co-transporter. The thick ascending segment also has a sodium hydrogen counter transport mechanism in its luminal cell membrane, which mediates sodium reabsorption and hydrogen secretion. Now let's revise these processes in this figure. This is a very important figure showing mechanisms of sodium chloride and potassium transport in the thick ascending loop of Henle. I repeat, in the thick ascending loop of Henle. The sodium-potassium pump in the basolateral membrane maintains a low intracellular sodium concentration and a negative electrical potential in the cell. The 1 sodium 2 chloride 1 potassium co-transporter in the luminal membrane transports these 3 ions from the renal tubular lumen, from the lumen of the renal tubule, into the epithelial cells lining the renal tubule. Using the potential energy released by the diffusion of sodium down an electrochemical gradient into the cells. Sodium is also transported into the tubular epithelial cell by sodium hydrogen counter transport. The positive charge of the tubular lumen relative to the interstitial fluid forces cations like magnesium and calcium to diffuse from the lumen of the renal tubule to the interstitial fluid via the paracellular pathway, as you can see in the upper part of the diagram. And you can also see in the lower part the names of the loop diuretics: furosemide, ethacrenic acid, and bumetanide, inhibiting the 1 sodium 2 chloride 1 potassium co-transporter protein. And as we just saw in the figure, the thick ascending segment, there is also paracellular absorption, reabsorption of cations such as magnesium, calcium, sodium, and chloride and potassium, as a result of the positive charge of the tubular lumen relative to the interstitial fluid. A very important point to keep in mind at this stage is that this thick ascending segment is almost completely impermeable to water. Therefore, most of the water delivered to this segment remains in the tubule despite reabsorption of large amounts of solute. Hence, the tubular fluid becomes very dilute as it flows toward the distal tubule. Now let's study the transport in the distal tubule. The first portion of the distal tubule forms the macula densa, which has its role in the autoregulation of the glomerular filtration rate. The next part of the distal tubule is highly convoluted and has many reabsorptive characteristics resembling thick ascending limb of loop of Henle. For this reason, it is also known as the diluting segment, as it dilutes the tubular fluid because it reabsorbs most of the ions but is permeable to water and urea. I repeat again, because it reabsorbs most of the ions but is impermeable to water and urea. In the early distal tubule, the sodium chloride co-transporter moves sodium chloride from the tubular lumen into the renal tubular epithelial cell, and sodium potassium ATPase pump transports sodium out of the cell across the basolateral membrane. Chloride diffuses out of the cell into the renal interstitial fluid through chloride channels in the basolateral membrane. The thiazide diuretics, which are used to treat disorders like hypertension and heart failure, inhibit the sodium chloride co-transporter. The figure shows the mechanism of sodium chloride transport in the early distal tubule. Sodium and chloride are transported from the renal tubule lumen into the renal epithelial cell by a co-transporter that is inhibited by the thiazide diuretics. And sodium is pumped out of the cell by sodium potassium pump, and chloride diffuses from the renal epithelial cell into the interstitial fluid via chloride channels. With this, I end today's lecture.